What if we could study melanoma not just cell by cell, but in the exact place those cells live inside the tumor? That's the promise of spatial biology. By combining spatial transcriptomics, single cell sequencing, and access to a valuable human melanoma biobank, researchers can get a more detailed look at how cancer behaves inside real tissue, how it interacts with the immune system, and why some tumors respond differently to treatment than others. May is Melanoma Awareness Month, making this the perfect time to spotlight how emerging genomic technologies are advancing our understanding of melanoma, tumor immunology, and the future of precision oncology. This is DNA Today, a podcast from Genepool Media where we explore the breakthroughs, challenges, and human impact of genetics and genomics. I'm your host, Kira Deneen, a genetic counselor and award-winning science podcaster and speaker. In this episode, we're joined by three experts. Professors Thomas Tüting and Andreas Braun are German dermatologists and researchers whose work focuses on melanoma, tumor immunology, and translational cancer research. Professor Tüting was recently awarded a major ERC grant supporting collaborative cancer immunotherapy research. We're also joined by Dr. Jia Hui Khoo, project manager at MGI, who brings expertise in spatial biology and the technologies helping to make all of this research possible. Together, we're discussing an exciting melanoma project profiling human melanoma samples from the University Hospital Magdeburg's valuable biobank and exploring how MGI's DCSP approach, spanning DNA, cell omics, spatial omics, and proteomics, can support translation in human pathology and oncology. Thank you all so much for coming on the show. It's always exciting to be able to talk to people in genetics in other countries. I'm coming from the United States, but we're talking to all of you in Germany. And I wanted to start laying out the conversation, Thomas, with you explaining the current landscape of melanoma research and treatment. And I'm thinking about particularly where immunology and immunotherapy fit into this, as we're going to get into.
As many of you might be aware of, the immunotherapy of cancer has largely been developed in last 2 or 3 decades using melanoma as a poster child and led to the Nobel Prize for the discovery of PD-1 and CTLA-4 blocking antibodies, which have been first approved for the treatment of melanoma and now are used widely against many other cancers. So one of the topics that are currently basically mostly studied is why do some patients respond very well to this treatment, whereas others resist. So it's basically trying to figure out the biology of resistance, which again happens in many other cancers, actually much more than in melanoma. So understanding response and resistance to immunologic treatments, the focus of a lot of labs over the world. And they're using more and more genetic techniques. You can barely skip an issue of Science, Nature, Cell without a paper on this. Just recently again. And most of it is now understanding how the genetics of cancer cells, so the genetic mutations, but also the epigenetic dysregulations, how they affect treatment responses. So this is where we are also trying to understand a tiny bit of the biology.
Yeah, it's very interesting just how this could potentially be really groundbreaking in terms of what we're learning. And Andreas, could you tell us a little bit more about this project and what makes it really exciting for your team?
Sure. Thanks for having us in the first place. So what we're trying to do in this project is we are trying to understand what determines how tumors respond to immunotherapy or whether they don't. And what is becoming more and more clear is that the interplay between tumor cells and the microenvironment plays a critical role here. And when I say microenvironment, I don't just mean the immune cells, but also other cells, non-cancer cells that are present in the tumor. One of the prime candidates that we are looking at in this project is endothelial cells, as they are let's call them highway, how immune cells enter the tumor. So the endothelial cells, this is one of the hypotheses, determine whether the tumor actually is visible to the immune system or not, and whether immune cells can enter the tumor or not. So the long run is trying to understand what drives the vasculature to either allow immune cells to go into the tumor or prohibits them from entering the tumor and thereby prohibits therapy responses to ultimately therapeutically manipulate it.
And it's really helpful. I'm learning that where you work, the University Hospital Magdeburg has a large biobank of human melanoma samples. So, Tomas, what makes these samples particularly valuable for this research you're doing? And how can access to these samples enhance our understanding of the tumor heterogeneity and differences in this treatment response that we've been hinting at?
I think the most important part of any sort of collection of biospecimens is the clinical annotation. Actually, we're not the largest center for melanoma, either in Germany or if you compare to other places, like, for example, in the States, they have much larger biobanks. But what we have in Germany as an advantage is that we see the patients when they first come with a primary melanoma in the skin. Frequently in the States, this is somewhere in a dermatologist setting, and then the immunotherapy happens in the medical doctor settings. So it's more difficult to get the whole story from the beginning towards the treatment towards the end. And we went to great lengths to clinically annotate our samples, and especially the primary tumors. They are valuable. There's only one. You only have a few chances to look at them. And so this is a little bit where we have a cutting edge. One other thing that I think is advantage in this extremely competitive field is that we also have a mouse model for the primary tumors where mice, which usually don't develop melanoma because of genetic alterations, do develop melanoma and do develop primary melanomas that are actually also initiated, accelerated by UV light. So UVB sunburning, UVB irradiation, which imitates the clinical situation a bit more closely.
This project is designed to profile about 50 stomics, spatial transcriptomics data alongside matched single cell data sets from these human melanoma and mouse models that you're referring to. So, Andreas, what are the main scientific questions that you hope this data set will maybe answer?
So what we do in routine clinic patient care is we see these tumors as dermatopathologists in H&E stainings, and we always wonder, what is it that these cells actually do? And you can tell a lot by the morphology. Everybody who does dermatopathology and especially melanoma pathology will know that the architecture and the spatial context of the lesion is essential for the diagnosis. But what our routine histopathology doesn't allow is in-depth characterization of phenotypes of these cells. So what we are hoping is that with the combination of stomics, so a spatial RNA sequencing technology, in combination with single-cell RNA sequencing, we hope to integrate these data sets to achieve an in-depth single-cell resolution on the transcriptomic side and anchor these in their spatial context. So we can then try and understand what these cells do, how they communicate, and potentially what drives immune cells to either enter the tumor and become active or don't.
It's interesting to be able to get so much data of the single cell and then put that all together. I think that's just such a interesting, different technology and technique that we couldn't do many years ago that now is one of the tools that we can pull out of the tool belt to use. Tomas, anything to add to this in terms of the kinds of translational insight that could come from your project?
Yeah, I was going to say there's one important aspect that Andreas maybe didn't emphasize enough. So what is becoming more and more clear in these analyses of the spatial context of cells is that cell identity is very strongly determined by where it sits and which tissue it sits. So this is why a lot of the more recent analyses published, I say weekly, focusing on what people call neighborhoods or niches. So basically, they don't look at a single cell in isolation. They look at the single cell in terms of who are next door neighbors and who basically orchestrate their state. There have been some seminal papers that, for example, for fibroblasts, but also for endothelial cells, which basically have a common origin, but they differ according to where they land after embryonic development. So an endothelial cell or a fibroblast, even in different places of the skin, differs. So it's different if you are on your scalp, where you have a lot of UV radiation, you become older, or whether you are on your palms and soles, or whether you are somewhere on the body. And then what intrigues me is that normally if you cut yourself or anything, the tissue will heal. And it will, again, later, every cell will know its position, right? Whereas in the cancer cell, which also frequently people say, oh, I sort of injured myself, it just didn't heal. So wounds that do not heal. So somehow cancer cells, due to their genetic perturbations that we don't understand yet, forget this anatomic knowledge where they are supposed to sit, who they are supposed to talk to. So then instead they just grow into a different tissue invasively or metastasize and set up shop in some other organs, right? And actually what controls this is not known. And we think that part of the mystery will be if we understand how the cells know where they are. So if they basically know how do they orchestrate their environment. So that I think we can unlock with spatial technologies and spatial transcriptomics, of course, like we do with the StereoSeq technology, is one possibility to understand what people in development call morphogen gradients that basically control your sort of who you are and where you are supposed to go. And we also think that during inflammatory responses, which are important in injury, that you also have gradients that somewhere intersect with the morphogen gradients. So we're somehow thinking about visualizing that, where I think especially the spatial sequencing-based StereoSeq technology will be helpful.
Yeah. And Gia, I have to bring you into this part of the conversation because you have such expertise in this type of technology. So anything to add in terms of what Tomas was sharing with how spatial omics is so helpful in understanding like where the cells are located and some of the genetic information that they're using to be able to figure out, oh, I'm going to go past the normal cell check, be, a rebel cell in terms of being cancerous with melanoma?
Thanks, Kira, for the question. I think overall, Thomas summarized it quite well, I would say. But I think just to highlight this point that I would say that if we look at the current scientific community, a lot of the researchers are still approaching spatial data, have, same previous side of analyzing single cell data sets or histopathology data sets. I think that now the community needs to really look at the technologies that's available, which I would think that, which is still quite recent. That's, how can you take on all these different layers of information? Now we can look at information such as genes, RNA, proteins, but also know where these cells are actually physically located within the tissue. So when researchers do the analysis, they will still think about it as if they're analyzing a single cell. So as mentioned by Thomas, they would be looking at fibroblasts, but they don't really think of where they're located in different tumor microenvironment. And then when they approach the analysis, they actually need to think about all these cell-cell communication and also interaction that are happening at the same time. So these all, I think, is a knowledge that the community is starting to develop. And then we as a platform provider are hoping to have more reference projects.
Yeah, it's incredible that MGI does have this technology so that they can help researchers like Andreas and Tomas be able to figure out this really complex information, because I think about years ago, we're like, okay, we'll learn the genetics of that cancer tumor. And it's not that simple. It's learning the heterogeneity of it, of, well, these cells are different in this way and have these mutations in those cells. And not just that information, but mapping it too is just so important. Not something I would have thought about years ago, of like the actual location really making a difference. And along with this, we were talking about these stomics technologies. Andreas, anything to add in terms of how these technologies have become useful over the years and how you see this continuing in your work?
So I'm actually, every time I think about this, I'm a little mind boggled, because I would never have expected to have this depth and this resolution of data at my fingertips at this point of time. So when we do the bioinformatics of our analysis, we always, when we did single cell, we always wondered, so how could we put these cells somewhere into context? And there were algorithms that tried to do that mathematically and infer which cells are alike, so which cells should be sitting closely together. But now actually having this basically supercharged, because now we have the spatial coordinates of these cells. So this is what always completely hits my nerve as a dermatopathologist and a researcher, that we have this spatial context of those in-depth analyses. I think for me, the next step is, how do we make sense of that? Because the data is becoming increasingly more complex. So it's very challenging to keep track of all the new tools and new analyses, the new algorithms popping up to actually analyze these and to actually make sense of what we see there. So this is, I think, what makes us very, very well suited in this project setup also, because we have the pathology background. So we are able to bring this morphologic sense and this clinical sense into these data sets and fuse these together to actually derive meaningful information out of this. Because at the end, we're still trying to understand cancer in order to treat patients better in the future. So this has to have some sort of meaning in the end, which I think we can provide with our project.
Certainly. And we've spent time talking about this spatial omics and how important it is to see like where these cells are and how they're different. But there's other different technologies that you're both using in your research. And so I think about MGI's DCSP approach, which stands for DNA, cell omics, spatial omics, and proteomics. So, Jia, tell me a little bit more about how using all of these tools is helpful. We don't talk about proteomics a lot on the show, I guess because we're called DNA today, but maybe I'll need to change the name of the show at some point to more encompass that genetics, genomics, all this research. We're going beyond DNA in a lot of senses. So tell me more about these different approaches and the technologies around them that help us create more of a complete picture of melanoma biology.
Yeah, so I would think that's a useful way to think about MGI's DCSP approach is that we're now trying to reconstruct the full biological system across multiple layers of information. I would say that each layer addresses a different biological question, and then together they are aligned with the flow of the central dogma, which is basically the central dogma tells us that, how genetic information is transcribed from DNA into messenger RNA and then translated into protein. So it tells us how DNA can be expressed as functional molecules that drive cellular behavior. So basically in this case, MGI, we are trying to make a series of innovation so that we can integrate DNA genomics, cell omics, spatial omics, proteomics, so that we gain a good understanding of these very complex biological systems. So if I take here in the context of melanoma, so Stereosig would fall under the spatial omics segment. So in this case, we are now generating a spatially resolved unbiased whole transcriptome map of the melanoma tumor. So this will now tell us how gene expression programs are organized into distinct tumor region. Are they invasive fronts? Are they, tumor excluded zone, hypoxia, etc. So this is all telling us that how the pattern of cell-cell interaction and also tumor microenvironment can change. So, and also I think because Stereosig is unbiased and also species agnostic, I think we have an advantage here that for Thomas and Andrea's project, they can easily switch between like human and mouse samples in the same assay. Then they don't have to redesign any targeted gene panel, which can be costly. And you may also miss some important target depending on the field that you want to study. And then for, just moving to proteomics, obviously, that for RNA and protein level, there are not two different layers of information. They are not always at the same level. But I would say they're equally important, or that the community has started to realize the importance. I think Andreas and I, we have been discussing about trying to integrate Stereosig with multiplex immunofluorescence or even CySeq, so that in this case, we can combine from maybe 3-4 proteins to over 100 protein targets within individual cell, so that we can now perform RNA and also protein co-analysis. So this will really help because not just it's 2 layers of information, but also with the protein information, it will help us to better distinguish the different cell types. So for instance, in endothelial cell, because the shape is weird and because as histopathologists, they know very well about the biomarkers or labeling the endothelial cells, they can now combine this and have a more accurate understanding of the entire tumor. And then just finally, for single cell analysis, we're keeping this in the project because they provide higher resolution or characterization of cellular identity and also these single cell states that can then afterwards complement the spatial omics data set. So I think that right now, spatial omics technology still cannot fully replace the single cell technology, which is why we're keeping them so that we have a very comprehensive data set. So I think ultimately we just to highlight that for MGI, it is one of the very few companies that is now having the capability to fully support the DCSP approach. We have the multiomic solution, which covers single cell, spatial, proteomics, but also now we are having the sequencing platform that enable us to generate these data set at a very large scale. So this is really important because then it means that the researchers can, generate all of the data sets, but they don't have to about the technical variation platform. Don't have to do a lot of optimization because then, from library prep to sequencing, because then these will all be provided in the same ecosystem.
Yeah, putting all of these technologies together and then all the data that comes out of it and layering it is really helping to create more of that complete picture. And it makes sense that we need to have different angles to be able to understand this better. Thomas, one thing I wanted to circle back to that we've mentioned earlier is that people respond differently. They'll have maybe the same type of specific cancer diagnosis and they get the same treatment. So those two people may respond differently. And that's really at the crux of what the research project is, to understand those differences. Can we use precision medicine to be better at saying, oh, this subtype needs to have this type of treatment? Have you learned anything yet from this research project in terms of figuring out some of those differences, or is this really just more ongoing than I'm alluding to?
We're still at the beginning. The challenge in the field generally is that, of course, cancer and therapy has dynamics. So cancer cells are moving targets. And of course, with a certain biopsy of the tissue, we have only a snapshot. So one advantage that the sequencing-based spatial approaches have is that you also can have an idea of the dynamics of transcription, because you can look a little bit into what is being produced right that minute, through different targeting bioinformatic techniques like RNA velocity and so forth. But in the end, we want to be able to assess better the dynamic responses of patients. And because right now most treatments basically are put on and then they are kept on, and how we dynamically respond with treatments, sort of how we adapt treatments to responses and so forth, and select then maybe alternative strategies and which ones would be suitable, that is still very much unclear. We hope to be able to make a little contribution to this project. But of course, many different labs and many different kind of cancers are looking at this. So we think that we have, by focusing on the endothelial cell, immune cell, tumor cell interplay in this what we call antigen presenting niche that happens in the tumor, we think that we have a sort of a little bit competitive edge, because we also have a mouse model for that, where we can experimentally perturb the system, right? Which is, if you only look at clinical samples, you can have a clinical trial and so forth. But then that is much more laborious to come to experimental, if you will, conclusions, because of course your trial and your drug is also an experimental intervention, if you will. So this is sort of a small hope, but the road is long. So I think at present we are very much at the beginning.
Yes, with so much science, and there's so much failure along the way, too, to eventually get to something that is groundbreaking, that really changes either on the diagnostic beginning aspect, the just different therapeutics, and everything in between. Andreas, any other thoughts that you have in terms of how this research could impact? I'm thinking could be like biomarker discovery to help with better diagnostics. this patient stratification of different types of treatments and just developing different types of treatments, because this type of research can have impacts in all of these different areas along the patient's journey?
Yeah, it's actually a great question. So I think one of the challenges of today is that we have either technologies that are rather high throughput, but don't give us this in-depth resolution that we have. I'm thinking more like routine H&E histopathology, immunohistochemistry, the bread and butter of the pathologist. And on the other end of the spectrum, we have these very highly resolved data that like we are trying to establish in this project, which obviously are way too tedious, too expensive to do for routine patient care for every case. The throughput just isn't at a scale that we could provide this cost effective to everybody in clinical routine patient care. So I think part of our aim in the long term is to bridge that gap, to identify both on the mechanistic level processes that govern immunotherapy response or non-response, to then experimentally permutate these processes and understand them better, so we find potential new ways to treat our patients. And on the other end, if we have these highly resolved data sets, we may be able to find markers that we can then pin down in maybe a simple H&E stain, or maybe in an immunohistochemistry for a specific protein, where all the very highly complex analysis brings us the knowledge that we then might use in routine patient care. So kind of a journey there and back again type of thing. So I think we have a twofold approach into bringing this research into benefit for the patient.
It's exciting just to see all the work that you've already done and the progress made using these technologies, but even more so, the tantalizing what is going to come out of this research. And I'm thinking about not just using these types of technologies, spatial omics, in melanoma and immunology research, but beyond. So I think it's just, this is a cool project to highlight, to show how these technologies can be used in research. But really tip of the iceberg in that sense of just how we can use this and how genetic research is really shifting, of needing all of these different technologies. We can't simply do DNA testing and sequencing and saying, okay, great, this is what we're working with. Like, that is one piece of the puzzle. So thank you so much, Tomas, Andreas, Jia, for coming on and exploring this with me and just seeing like what technology is out there, how are researchers using this around the world. It's been cool that we've been able to do a lot of episodes with MGI of just different countries. It's like, today we're in Germany. Where are we going next? So thank you all so much for taking the time and explaining and really just exploring all of this with me.
It was a pleasure to be here.
Thank you.
That's a wrap on this episode of DNA Today. To keep exploring the world of genetics and genomics with us, head to DNAtoday.com for hundreds of episodes, show notes, and resources from our archive dating back to 2012. DNA Today is part of Genepool Media, the Science Podcast Network. To check out more of our episodes, visit genepoolmedia.com. For questions, guest pitches, or sponsorship inquiries, email info@dnatoday.com. And if you enjoyed this episode, a rating or review on Apple Podcasts or Spotify helps more people find the show. Thanks for joining us. I'm Kira Deneen, and I'll be back next week to continue discovering new advances in the world of genetics.